Thank you, Josh, for organizing this really nice program and a lot of exciting stuff in the whole day. So, because I'm a biostatistician, my talk is really, I cannot give a talk without data. So my talk is really about how to use the large public or EHR data for clinical ophthalmology research. And so I will talk about three different types of data which I navigated in the last 2 years, include the Alpha-Us database and the TriNetX database and the AI-ready database. And my talk, I will give each database, I will give one example how we do the research. And I also will give a little tip how to do the best research using all this data. So first is the Alpha-Us database, and Alpha-Us is really NIH-sponsored research. Started 2018, and in 2019 they have a paper in the New England Journal of Medicine, and they talk about the rationale for this research and what's data available and what kind of information is useful for the research. So everyone want to use this data, I encourage you to read this paper. And the Alpha-Us had to enroll about 1,000 patients and 1 million patients across the United States, and is one feature is the population is included a lot is underrepresented population, unlike the other study, and it's really good for use study for a large scale research and genomic and disability research. And currently we have 881,000 participants already enrolled in the study. And the Alpha-Us really have field-rich data, include the EHR data and the genomic data and survey data, which I will go a little deeper in the next slide, and also wearable device digital health data. The bad thing is not everyone have all the data, and you can see the number of patients have different data components is different. So this have to be careful when you use the data for the research. And the good feature about the Alpha-Us, actually they have website, and you can search the keyword. For example, if you want to study one disease, and you can type in the keyword and get some idea how many patients available for you to do the research. So the survey data is really one, I feel is a unique feature of Alpha-Us database, and it's pretty rich, include the basic survey data, include the demographic and social economic status and overall health and lifestyle, healthcare utilization, and social determinant factor and mental health. And also because this survey data use at a different stage of the study, and the number of patients have data is different. The Alpha-Us is really field-rich data, and it's Alpha-Us a huge opportunity to do the clinical research. And they have one paper, Nature Medicine, and this paper was led by all the different institute under NIH director, and include the National Eye Institute, Michael Chang, most of all you know. And basically this paper really highly recommend or promote use of Alpha-Us data for the clinical research. And they talk about 9 different areas you can use this data for the research, include how the genomic association with the disease, or AI model, or develop prediction model. And I will not go every field, but if you want to use Alpha-Us, I recommend you read this paper. You may find some idea and use the data for your research. And people already use this data for many kinds of research, not eye disease research. And they have a website. You can find all the publication from this database. So currently already over 1,000 publications. And also in the website, you can find all the research proposal people posted in this website. So if you want to do something, it's good to check the website to see with the research question you want to do is already people already started work on, so you don't do the same thing. So we have used this data in the past 2 years, and we work on many different projects, and some of the projects already lead to the publication, and some is presented in the professional conference, include the AVO, and all this project is really, I work with excellent students from Penn, from Jackson, and a few other collaboration researchers. So, and you can see this research was recognized by received some award, including Han Yu and my students work with me for many projects and received still fine ophthalmology medical student research prize, and also KD received the Penn Medicine Alumni Fellowship and Public and Global Health Policy Award. And just last week, Kevin and Song work with a project on the metabolic syndrome with the risk of different eye disease and receive the AVO travel grant. So I will not go each detail for each project, but I want to highlight one project, which is really the first project I work with the user of us with Alan Zhao and to study how the dry eye disease associated with the mental health condition. So this is really a cross-section study, and we use the snow code to identify the dry eye disease, including dry eye, meibomian gland dysfunction, or tear film insufficiency. And so this is our case, and the control is really we do a 1, 2, 3 match to find the patient who don't have a dry eye disease. And because it's always important to control the confounding factors, we use the propensity score matching, include the demographic factor and some other social determinant factor. And in the model, we also adjust for a few comorbidities. So we look at all the different mental health outcomes, and these results, you can see the patient who have dry eye, they have almost 3 times a higher risk of any mental health condition. And for the CPC mental health condition, you can see the odds ratio are above 2 or 3. So this indicates the patient who have dry eye, they're more likely to have at a higher risk of the different mental health condition. And interestingly, we also found the black patient who have dry eye is have higher association than the white patients. I did not have show all the detailed results. So the next database I want to talk about is the TriNetX. This database we just recently started work on in 1 year or above. And the TriNetX is really a global federated database. Actually, it have 2 different databases. One is called Global TriNetX. It includes almost over 300 million patients from over 200 healthcare organizations. And we also have a US TriNetX, which have about 170 million patients, include 86 health organizations. And this data is really real live data. Basically, every time we go to the database, the number certainly is updated, may not be the same as the last run you run. And one nice feature of TriNetX database is really they have called analyze platform. You don't need to write a code. Basically, you just go to website and you build the cohort and you select outcome measurement and you select exposure variable, and the program will run for you. So you get all the results. You don't need to write any code. This is good thing for some people, but maybe also bad thing for the research, because the people who don't have a good knowledge on how to do the statistical stuff, or how to don't have clinical knowledge, may not use the data, analyze data right, and the results may not be reproducible. So this we have to be very, very careful. So the TriNetX is also very rich, include demographic data, diagnosis, procedure, and medication, and they also have some limited lab test results, and include genomic data also. And the people already use the TriNetX for many different research, and not just for eye research. And you can see this publication I found in a search in the PubMed, and you can see the number of publication is really increased in the past 3 or 4 years. You can see even last year they have total about 1,600 publications, and this year we already have over 1,000 publications. So people do use this TriNetX there for many kinds of research. And we also work on a few projects and highlighted here. And And Hanno worked with me for one, I feel is really interesting project. We look compare the laser with antiviral treatment for RP, with they have a different impact on the neural development. So, and this research Hanno received a travel grant and presented a post meeting the 3 months ago. And we also have a few other projects. I will not go detail, but it's all work with the paramedic student and my collaborators. So I want to highlight this project for the neural development, how antiviral have impact or not have impact on the neural development. So they do have some, when the antiviral become available for treat RP, a lot of doctors do have a concern with the antiviral can be have impact on this premature infants, that neural development. And they do have a few small size study found that they have impact, but they some have other studies say no impact. And they have one meta-analysis a few years ago. They showed full data from many small studies, and they found no impact. So we feel this is a good opportunity because China database is huge, and we want to use all available data. We want to study with antiviral to really have any impact on the neural development in this premature infants with RP. So we do data use the 2011 to 2025 because this 2011 is the time when the antiviral medication become available for treat the RP, and we find use the procedure code to identify the laser treat RP or antiviral treat for RP. And we do use the one-to-one propensity score matching to control all the possible confounding factors, including the demographic factor and a lot of comorbidity factors. And we use the time to event survival analysis to compare the neural development after the treatment. So this result, we found the antiviral really have no impact on the neural development, and include the hearing loss, any development delay or motor delay or cognitive delay, and the speech delay or cerebral palsy. Actually, for this cognitive delay, is the laser treatment have a little higher risk than antiviral treatment. So the third database I want to mention about is called AI Ready. This is really NIH funded study. Want to build a flagship for use this data for the AI or machine learning model database for validation or development. And the aim of this study is enroll 4,000 patients, and from 3 United States, including Birmingham, Alabama, and San Diego or Seattle. And this database really include patients who have a different stage of diabetes, include the control, health control, no diabetes, and the pre-diabetes, and the diabetes with oral need oral medication, and the diabetes need the insulin treatment, and they cover different race, ethnicity, and the goal is 4,000. Currently, they have over 2,000 patients already enrolled, and the data is available for us to use, and you can download the data from this website. And the database, the AI database is one unique feature I feel is really useful for ophthalmology research because they have a few rich image data, include the fund photo, OCT, OCTA, and they do have also have a very complete systematic clinical data and wearable physical measurement. And another feature is really I like it because they do have a cognitive function measured by the MoCA score, which we take advantage and do the research. So these are 2 projects we already work on, which lead to the publication. One is the work with the chemist, our medical student, work how with the diabetes severity is associated with the cognitive function, which we published already BNG open. And another project which I will highlight in the next few slides is I work with the Mengyu and Dr. Ben King, and to use the deep learning model for calculate retinal age, then to study with the retinal age is associated with the cognitive function measured by the MoCA score. And this paper, actually yesterday, we just accepted for the publication in the ophthalmology science. So I want to talk a little bit about retinal age, and in the front photo, or retinal images, they have a lot of information, and include people already report how the retinal age can be used for detect systematic disease and include the cognitive function. So we want to look at retinal age, how associated with the cognitive function. And the Australia group, they develop AI model. Basically, you feed in the retinal, find the photo, and the output, the AI model can give you the estimate for the retinal age. And they have a model developed based on the UK Biobank and also validated by use the data from Chinese cohort. And the idea of a retinal age calculation is based on the really based on the physical feature in the found photo. So we apply this the AI model, and from the Australia group, they nicely share with their code, and mainly use this model to calculate the retinal age from all the participants from the AI-ready database. So this example of the retinal age we calculate, and the left side, this image shows the retinal age is younger than the chronic age. This person must have a healthy lifestyle. And the middle image is really the chronic age and the retinal age. They are similar. And the right side is the retinal age is older, which you don't want to be because they indicate that you are beyond your chronic age, which is not good. So we look at how the retinal age associates with the cognitive function, and first we look at how the chronic age associates the cognitive function measured by the MoCA score, and the correlation coefficient is I would say it's mild, it's minus 0.2. This means when you get older, your cognitive function become worse, which makes sense. And we also look at how the retinal age associated with the cognitive function. We can see the correlation become much stronger, which is minus 0.4, compared with the chronic age, and we can clearly see the almost a dose response, clearly dose response relationship. When you get older, your cognitive function is better and the MoCA score is lower. And we also look at how with the retinal age can be used to kind of detect the cognitive impairment, which we defined based on the MoCA score lower than 26. And we can see the AUC curve is a measurement for how good the model detects the cognitive impairment. And you can see chronic age alone, the AUC is very low, only 0.6 something. And if we use the retinal age, the AUC is 0.83. The maximum is 1, so it's still good. And if we combine the retinal age with the chronic age together, you can get an AUC 0.87 something. And also you can see the percentage with the cognitive impairment is increased by the quartile of retinal age. So this supports the retinal age is have a much better than the chronic age to detect the cognitive impairment. So I cover all of us database, the TriNet database, and the AI-ready database. I think each database have their unique feature, and it really offers us a huge opportunity to do the clinical research. I feel all of us really, one feature is really they have a very good the survey data, and we can explore, and you can see how the social determinant factor can associate the different eye disease. And the TriNet ACE database is really, because it's huge, you can use this to study rare disease, and you can study how the one procedure compare with another procedure for treat different eye disease. And the AI-ready database is unique because they have a very rich retinal image data. You can use this data for development the AI model or validation the AI model. But this database also have some limitation we have to be aware of. And because all the diagnosis of a disease is based on ICD code or billing code, which can have some misclassification as we know. And the TriNet X database, I already mentioned about is really the nice thing. You can use go to the database, do everything by yourself without the need to write any code and without the need of consultation with the biostatistician. But the results may not be easily reproducible. You have to be very careful. So I always encourage people work together and make the results more reproducible. And also the database have some statistical challenge because of the misclassification, confounding, which is a big issue. Use this all different EHR database, and also missing data, and when you study many different outcomes, they have multiple testing issues, and patients have different length follow-up. So they do have some statistical challenge, and it would be nice to develop some more complicated statistical model to handle this different limitation of the database. So I want to give you a few tips based on my navigation with my student in the past 2 years. One recommendation I feel really important is really when you do a research, use this large database, always include the clinician, ophthalmologist, and also include the biostatistician or epidemiologist work together, because everyone contributes different thing. And because this clinical research, we do need to do the research in the context of the clinical care. And the data is not simple. You don't need a biostatistician to give you the guidance on how to analyze data in a proper way. And also, before you really do the research, I really encourage you to write the proposal clearly, define what's the analyzed cohort you want to study, and what's the outcome, what's the exposure variable, what's the inclusion criteria, and what's the confounding factor you need to adjust for. And this all have to be laid out before you do the statistical analysis. And also, it's highly important to adjust all the possible confounding factor when you do the comparison, because this EHR data can be easily confounded by a lot of different things. And first, you need to identify what's the possible confounder. And the next thing is how you apply different model to adjust for this different confounder, include use a motivator regulation model, or use a propensity matching, or use a more advanced approach like the causal inference. And also, it'd be good to include the positive negative control if possible, so that you can validate with the cohort you build up is really make sense. Because if you use a positive control, you can confirm with the association can be validated. And if you use a negative control, you can see with they have any hidden bias or residual confounding. And I also highly recommend do the validation by another person. This is hugely important for the genetic database. So I really want to acknowledge all my wonderful students from Penn and from Drexel. And in the past 2 years, I really enjoy working with all of them. I list all their photos. A few students I may not have included because I submitted the slides earlier. And I also really enjoy with Minji, and he's in audience, I believe, and for this wonderful AI retinal age research project. And I also want to thank you, my biostatistician and Jocelyn work all the project with support students for use of us database, and Ying provide a lot of guidance for my students on how to use the TriNet and also do the independent validation. And all the faculty collaborator, I really learn a lot from all my clinician collaborators, because I'm not a clinician. I really want to do the research in the context of clinical research. And I also thank you the full fishing program to support outline my research and research plan plannings. And finally, for my indoor chair fund, which I really use for my students to go to the conference, different meeting, and so that they can present their finding, share with the community. So that's it. Thank you very much for your attention.